Welcome back. We're going to get right into cell division. So cell division is also known as cell replication. And there are two major periods in a cell's life cycle. The first is called interphase, and this is a phase where basic metabolic activities occur, so maintaining life. And then you have the cell division phase where the cells actually divide. I love this picture. I found it on Google Images. It's just adorbs. So I use it. Hello, there we go. So this is a beautiful picture. This is not from your textbook, but it shows the two major groups of, or two major phases of the cell cycle. So we have interphase. Interphase is where your cell is going through metabolic activities. And so this whole region out here where you see G1, S, and G2, that's all interphase. So it's the majority of the cell's life cycle is in this interphase where basic metabolic activities occur. And then we have this very short period called the mitotic phase where you have cell division occurring. So during interphase, let me just go back for a quick second. During interphase, we go through three separate periods, G1, S, and G2. So this G stands for gap. So this is the first gap phase. Gap 1 is where proteins for DNA synthesis are going to be produced. The cell is going to undergo normal metabolic activities also. Then we have the S phase here. This is where DNA synthesis occurs. So S stands for synthesis. And so we replicate our DNA in this phase. In G2, we check to make sure that the DNA has been synthesized properly, and we produce proteins for the mitotic phase. We are also doubling our organelles and getting ready for mitosis. So what happens with DNA replication? If you took a general biology class in high school, or if you've taken general biology in the past here at college, then you already know that replication of DNA occurs before a cell divides. The reason we have to have this done is because all cells have to have that DNA in it. DNA is the blueprints or the genetic material that tells our cells what to do. It's kind of like the brain of the cell. And so during DNA replication, we're replicating every single chromosome. The process starts with DNA uncoiling. So DNA is a double-stranded molecule. It's going to unwind and unzip. So unzipping means hydrogen bonds between the nucleotides are going to break. The nucleotides come apart, and then DNA polymerase, which is an enzyme, binds and allows new complementary nucleotides on each of the parent strands to come along. And so remember that my dog's apologies. Adenine always binds with thymine. Guanine always binds with cytosine. So when DNA polymerase moves along, if there's a T in the parent strand, DNA polymerase allows an A to attach. If there's an A T will attach. If there's a C, a G will attach. And if there's a G, a C will attach. This occurs on both strands of DNA. So here it's unwinding. And here you have DNA polymerase moving along, allowing for new nucleotides to attach. So what we end up with is two identical double-stranded DNA molecules, each consisting of one of the old strands of DNA and a new strand of DNA. And because of that, it's termed a semi-conservative process. And this again is from our OpenStax textbook that I use for another class. I just love this picture. It's beautiful. It kind of shows what's going on. You can, I think, see, well, yeah, you definitely can see with the arrows that the DNA polymerase moves in the opposite direction. So one strand, it's moving this direction, and the other strand, it's moving in this direction. It's because you can only attach DNA to one side of the molecule. So nucleotides can only attach on a 3' end of the DNA. Oops, apologies. Hold on.
Grammarly does more than catch errors.
Sorry. That's not what I wanted to do. There we go. And so here's your figure that shows the same thing. You can see nucleotides are coming in and attaching on both strands. That's DNA polymerase's function. It allows those nucleotides to come in and attach, and new hydrogen bonds form between the nucleotides. So you get two identical daughter strands from one parent strand. So mitosis in a nutshell then is division of the nucleus. And this is going to produce two nuclei within the cell. Cytokinesis then is division of the cytoplasm, and it occurs right around the ending of anaphase, and it finishes at the end of telophase. And so in this, you have two daughter cells. Each of those cells will get one of those nuclei. So for mitosis, there are four basic phases that occur. So we have prophase, metaphase, anaphase, and telophase. In prophase, a lot of times textbooks will split it up into early and late prophase, but it's all prophase. So I usually say just prophase. You're going to have the nuclear envelope start to break down. The chromosomes, the chromatin, which is the uncoiled form, will coil up into the chromosomes, and sister chromatids will attach together. So each sister chromatid consists of one strand of DNA, or the identical strand of DNA. So they remember, your DNA replicated, so you have two identical strands. They connect at a region called the centromere. So that's this region right here, or here on this one, okay? Centrioles will start moving to the opposite poles of the cell, and spindle fibers will be formed. These spindle fibers are going to move and attach at the centromeres on each side. And so here you can see spindle fibers moving in, and they're attaching. And so you have a spindle fiber from each side, from each of the centrioles, coming out and attaching at that centromere positioning. These spindle fibers then puppet the chromosomes. They move those chromosomes around. So in metaphase, all of the chromosomes line up. The spindle fibers move them until they are lined up one on top of another along an imaginary plate called the metaphyseal plate. So it's along the middle of the cell. In anaphase, the spindle fibers pull apart the sister chromatids. And so you get one chromosome of each type moving to the opposite poles of the cell. This is also when the cytokinesis will start. So telophase occurs, the chromosomes will uncondense, go back into their chromatin or relaxed form. The spindle fibers break down, new nuclear envelope reforms around the chromosomes, producing a new nucleus. And then you have this cleavage furrow occurring. This is that cytokinesis that actually closes, or that actually splits the cell. So you have two cells, two daughter cells from one parent cell. And so cytokinesis is what produces the two daughter cells because it breaks the cells into two sides or two halves. This produces two identical daughter cells. This entire process produces two identical daughter cells from one parent cell. So in general, mitosis and cytokinesis occur together, but there are some examples where you may have the mitosis occur, but no cytokinesis. So that would occur in cells that are bi or multinucleate. Cells, the most common cell that I show people is skeletal muscle is multinucleate. It has multiple nuclei for each single muscle fiber. Cardiac muscle can be binucleate, and liver muscle can be binucleate as well. And I'll show you pictures when we get into the tissue portion. So this is the entire process, starting with the early, or this is interphase, and then we have early prophase, late prophase, metaphase, anaphase, and telophase. So I'm not going to show this video. That would be, I think it would not show up properly. So at this point, I would stop this and then watch your video. You have this PowerPoint posted, so you can watch this video. You just click on this animation here, watch the video, and then come back to the PowerPoint or come back to the videos in Edpuzzle, or if you're watching it on YouTube, okay? So you should be able to, at this point, answer these questions in your study guide. What are the three stages of interphase? So you're looking at that G1, S, and G2, and what occurs during those steps as well. So you know when certain things happen. What are the four basic steps of mitosis? Prophase, metaphase, anaphase, telophase. What is cytokinesis? Division of the cytoplasm. How does DNA get replicated? So that's up in this S phase, synthesis. When in the cell cycle does DNA replication occur? So that was vague. Does this occur? DNA replication occurs during that S phase of interphase. And what are the major events of each stage of the cell cycle? So what's going on in G1, S, G2, and mitosis, prophase, metaphase, anaphase, and telophase? Okay, I am going to stop here and we'll get into protein synthesis in our fourth video. Have a great day, and I'll see you shortly.